Alright, y'all, so today we're going to be talking more about speciation. So last time we talked about how speciation can happen either in different geographical locations, so that is by allopatry through allopatry, and it can happen by a single population splitting into two, so it's vicariance, or it can happen by a population, just a few individuals from a population going to a new area. So remember, when it's a whole new area where there isn't any other individuals of that species, that could lead to speciation. Versus if we have individuals going back and forth between populations, that's gene flow that we learned about earlier this week, okay? The other way that speciation can happen is through genetic changes first, right? So we could have disruptive selection, and that disruptive selection is acting on the genetics that are present in the population, where it's good to be at the extremes, right? Or through ploidy changes. So something happens wrong during a process called meiosis that we'll be talking about next week, where things don't divide properly, okay? And so either of those ways could result in speciation. And the question now becomes, when do we call two things separate, okay? So if we have two populations living in different areas, that doesn't mean that they're different species, right? So the population of, I don't know, cicadas living here, right, might be the same as a species as a population that lives in Richmond or something like that, right? So just keep that in mind, is that just because we might have some geographical separation of some kind doesn't mean that we immediately have speciation, especially for a species that's kind of long-lived like cicadas, where they have those long life cycles, okay? So let's go ahead and start talking about different species concepts. There are lots of them, and if you get enough evolutionary biologists together, they're all going to tell you their own favorite one, okay? But there's three that we're going to talk about today because each of them have pros and cons and can kind of be tied in together sometimes, or sometimes we just have to use one because it's the only one that we have available to us. So we'll talk about three different species concepts today. So let's start a new note. Okay, so let's start. Our new note is going to be day 15. And this is speciation concepts, or species concepts. Okay, and so there's going to be three of them. So we're going to have the biological species concept, okay, we're going to have the morphological species concept, and we're going to have the phylogenetic. Okay, and we can ask a question with each of these. So for the biological species concept, our question is, can individuals of two populations mate and produce viable offspring? Okay, so that's the question that we're asking with the biological species concept. With the morphological species concept, the question that we're asking is, do individuals of two populations look different? Okay, and the phylogenetic species concept is, do individuals of two populations always separate into different clades? Right, so remember clades from our phylogenetic trees, right? So we want different clades on our phylogeny. For the morphological species concept, we care about the looks. And for the biological species concept, we care about ability to make viable offspring, okay? So these are our three main concepts that we're gonna be talking about, and and the questions that we're asking with them, okay? So let's talk a little bit more about the biological species concept. I'm going to shorten it to the BSC, okay? So the BSC, we could have different types of what we call reproductive isolation, okay? So we have populations are different species if they cannot make viable offspring. Okay, so if they cannot make viable offspring, and this can happen in a few different ways. So it could happen, and we call this reproductive isolation. Okay, so this could happen through prezygotic isolation. Okay, so in prezygotic isolation, we never form a gamete, right? It's before the zygote is formed. And this could happen because we have different habitats, right? So if one is always on the top of a mountain and the other one is always in a pond at the bottom of a valley, they're never going to have a habitat overlap and be able to meet to form a zygote, right? So we could have different habitats, we could have different behaviors, right? So if this is really common in like something like birds, right? Where if the bird song doesn't match what a female wants, then she's not even going to consider a male bird as a potential mate, right? So we have different behaviors. We also have temporal isolation, so the gametes are out at different times. Right, so this could be breeding timing with, like, think about like rabbits maybe, if there's different times of year where they're going to be breeding. Or it could also be something like the trees and plants around us that have the pollen, and just the pollen is out at a different time as the flowers, right, of a different tree, okay? We could also have mechanical isolation. And this is where parts don't fit together. Right? So this is actually a pretty common one in flies, where flies of one species actually don't fit into the flies of another species, okay? So just the sexual organs themselves don't fit together, okay? So we could have prezygotic isolation, or we could have postzygotic. So here we've formed, what am I saying before the gamete is formed? Over here, sorry, this is a typo. I don't know what I was saying before a zygote is formed. Okay, postzygotic is after, hopefully you guys will change that, okay? After zygote is formed, okay? And so one of the biggest ones here is actually hybrid inviability. Okay, where the hybrid, when we have two different species mate, form a zygote, and the hybrid is not viable, that is, it can't survive, right? We could also have hybrid sterility. So when we have hybrid sterility, it's when you can make a hybrid, it can survive, but then it can't actually produce viable gametes, okay? This happens a lot, actually. This hybrid sterility happens a lot in the case of the ploidy changes, where the hybrid is going to be sterile, okay? So these are some different types of isolation that we could have with a biological species concept. So in a biological species concept, remember, populations are different species if they cannot make viable offspring, okay? And this is called reproductive isolation, okay? The next one that we were going to be talking about is the morphological species concept. Okay, so here, if they look different, it's pretty simple. If they look different, they are different. Okay. This though, a little thing to note about this is that this is highly subjective. Okay, so I actually really love plants. I didn't always love plants, but I really love plants now. And if I come out here to the garden, I'm just looking right now at one of my squashes that I don't know what it is at the moment. I don't know if it's a butternut squash or something else, if it's a pumpkin. It just came up voluntarily with the compost that we added to the soil. There's another one across the way, and I don't know if those two are the same species or different species right now, but if you ask Dr. McClutchy, who's in the department, he would be able to pop out here and tell me immediately that they look different, when to me they look exactly the same at the moment. Okay, but at the same time, if I'm looking at this, like the leaf shape and stuff, they look exactly the same. If I'm looking at the one that is further developed and has little tendrils forming, but the other one doesn't, maybe I would say, oh no, they're different because this one has tendrils and that one doesn't. So I'll just kind of show you guys real quick what I'm talking about. So here I've got this squash of some kind, and you can see these little tendrils forming, right? There's one over across the way. Where did it go? My garden is a mess right now. That doesn't have those little tendrils forming. So the leaves look the same to me, and I would be tempted to say that they're the same thing, and we'll find out in a few weeks whether or not they are the same. But here's the other one, right? And there's no tendrils. So maybe I would say, oh, they're different because one has tendrils and the other one doesn't, right? But maybe they're just at different stages of development. And so it's not always a great proxy for whether or not something is the same species or not, okay? And then the last one, okay, is the phylogenetic species concept. And this one typically relies on DNA whenever we can, okay, to make phylogenies, right? And so the idea is, if all individuals from one species is in this clade, or population, I should say, and all the individuals from another population are in this clade, then some people would count that as two separate species, right? But maybe if we had sampled even more individuals, I'm gonna switch one of these colors. If we were able to sample even more individuals, maybe we would find out that, oh, there is actually an individual from species population 1 down here, or maybe if we did more sampling, we would see that there's some hybrids or something like that, where there's some individuals in both clades, right? So maybe we have different amounts of sampling would tell us additional information or something like that, okay? So this can also be a little bit murky on where to draw the lines, right? The biological species concept is a pretty straightforward question. Can they make viable offspring or not, right? But there's pros and cons to this as well. So let's just do a little chart of pros and cons for these, okay? So pros, cons, why am I putting them so close together? Cons. Okay, so we've got the biological species concept, right? And remember, it's asking viable offspring. Right? The pros are that it is very direct, yes, no question, right? But the cons are it's limited to species that reproduce sexually, right? So if we have species like bacteria, we can't do it, right? They just divide by binary fission, right? Can't use it on bacteria or archaea or anything that's extinct like dinosaurs, right? We can't use this. Also, if it has a really, really long lifespan and reproduction time, it's really hard as well. So like the cicadas that are out right now, it's really hard to figure out if things are separate species using the biological species concept, because it takes 13 to 17 years for a lot of these different species to come out. So if they're coming out at the same time, maybe we're like, oh, they seem like they are the same, right? But if there's two sets of two different broods coming out at the same time, then it's a little bit harder to kind of make that call, right? Because we just can't apply this concept. So the next one is morphological species concept, and this is how do they look, right? So the pros are that we can use this on bacteria, so broadly applicable, right? So we can use it on bacteria, archaea, right? So both prokaryotes, right? Extinct species, etc. But the cons is that it's highly subjective, right? It's really subjective, and it's subjected to our senses, right? So our senses of, our five senses of like taste and touch and sight and sound, right? We can use those senses, but maybe trees have a different way of another sense that we don't have that they can use as well, okay? Or that we don't know about. So it's highly subjective is the major con. And then the last one is the phylogenetic species concept, and this is asking about the clades and if they're in the smallest clade or not. So the pros is if we have DNA, so if we can get the DNA, we can apply it, right? If we have the DNA, we can apply it, and we can look to see if it's the smallest clade or not. But even that has some subjectivity, and it also is really important with sampling size. So it's really a sampling size issue. If you only get like 5 individuals from these 2 different populations, then you might not have a good enough ability to determine whether or not they're actually different species or not, okay? So sampling size can be a major con to this. There is some subjectivity to it as well sometimes. And so a lot of times, if you can, it's best if you can kind of mix these species concepts together. And some people do that a lot, where they've made up names. There's like 40 plus species concepts, where there's some that do different combinations of these as well, okay? As well as other ones that focus on different things, okay? Can have issues and etc. Okay, so with the morphological species concept and the phylogenetic species concept, you can over or underestimate the actual number of species, right? For both the PSC and the MSC, right? It's easy to over or underestimate. The PSC, this last one, is getting better as our ability to sequence gets better. We're able to have higher sample sizes and being able to sample more, because it's just cheaper and more available and easier for us to do it, and do whole genomes. If we're just sampling one gene or a few genes, in the past this has really misled us, okay? And that's why people use a combination of these pretty frequently. So again, going back to those sawflies that you guys had done that case study on, it's real research that has gone on in the bio department at UK. And I worked in Dr. Lennon's lab, and when we just published our paper on the eastern species, including Lecontium pineatum, but a whole bunch of other ones too, 19 different species, we kind of used a mixture of these. We talked about some morphological things. We did do phylogenetic trees for them, and then we have some information about which species can produce viable offspring or not, and we kind of tied all of those together when we actually wrote the 2022 paper. I guess I was working on it in 2022. I don't know what year it got published. Okay, so just kind of note that we have these different species concepts, and that the ways that we can get to these different species, right, regardless of which concept we're applying, we're always going to have those different ways of allopatric speciation versus sympatric Where in allopatric, you have geographic separation first. And then genetic differences accumulate. And in sympatric speciation, you have genetic differences first. Okay, and then other additional things can follow that. Okay, in allopatric, you could have vicariance, where a single population gets split, or you can have dispersal, where individuals from a population go to form a new one. Okay, in sympatric speciation, you're going to be in the same place, and you can either have disruptive selection, right, or you could have ploidy change. Something like first you might start from 2N to 4N, and then maybe it changes from there. Okay. All right, so let's take a look at our learning objectives. I just wanted to do a quick review of that because I know some people struggle with it sometimes. Okay. Okay, so here's our speciation. Okay, so learning objectives. So here, be able to compare and contrast the biological species concept, the phylogenetic species concept, and the morphological species concept. So I want you to be able to like tell me which of these questions they ask, right? What is going to kind of like solidify, including what type of biological species concept might be at play, whether it's pre-zygotic isolation or post-zygotic isolation. Looking at the morphological species concept and some of the pros and cons of these three different concepts that we listed out here. Okay, be able to tell me those things. Contrast the effects of gene flow and reproductive isolation on the evolution of species. Yes, so we have barely talked about this. So here, if you have something where you have vicariance happen, right? Oops. So if you have something like vicariance happen, but then you have gene flow happening, then we're going to make it so that we don't have new species. So let's just look at this really quick. So here, if we have a population that's split by vicariance, right? So that there's two different populations, right? Do those green dots. Maybe over here there's actually green and yellow, right? And let's say that there's two different ponds, but there is actually a little hole somewhere. Oops, back. There's actually a little hole somewhere where individuals can flow back and forth, right? So maybe every once in a while, right, individuals can go back and forth in this pond. So this yellow one is over here now, and this green one is over here now. And so this right here is an example of gene flow, right? And when we have gene flow happening, it is going to prevent speciation, right? Because remember from a few days ago, gene flow is going to make two populations more similar, okay? So gene flow will prevent speciation, right? Versus if we have reproductive isolation, even if we have gene flow, but these two can never mate with each other, then it's fine, right? And we have reproductive isolation. So reproductive isolation kind of helps speciation happen. Okay. And remember again that we have reproductive isolation can happen at multiple levels, right? So it could be pre-zygotic or post-zygotic, right? And either way, it helps speciation go along a little faster. Just want to make a note, pre-zygotic or post-zygotic. Okay, the next one is compare and contrast pre-zygotic and post-zygotic isolation mechanisms and be able to give examples and state the reproductive isolation occurring, okay? So if you go back, oh, not to this day, oh yeah, right here, to pre-zygotic and post-zygotic isolation. Remember, pre is before the zygote forms, post is after the zygote forms, and different isolation mechanisms can happen. So different habitats is habitat isolation, behavioral isolation. This is temporal isolation. I didn't actually name it. Temporal isolation, or mechanical isolation, where the parts just don't fit together, right? Versus post-zygotic isolation, we have hybrid inviability and hybrid sterility, okay, are the two ones that I want you guys to be focused on, okay? All right, and so if I give you an example of that, I want you to be able to tell me, if I tell you this cicada is out right now in the year 2025 in June, but there's another cicada population that comes out in August of 2025, you should be able to tell me that there seems to be some temporal isolation happening there, where the gametes are out, the adults are out and singing at different times. Okay. Let's see, what was that one? Number 3. And then number 4 is compare and contrast geographic isolation by vicariance versus dispersal. So remember that from yesterday, where we had vicariance is where one population divides into 2, versus dispersal is where some individuals of one population go to a new location. And that is a little bit different than gene flow because it's a whole new location where no individuals currently are at, okay? And then the last one, given a sequence of events involved in allopatric or sympatric speciation, arrange them in their correct order. So remember up here where we just had that reminder that allopatric speciation, geographical separation happens first and then genetic differences accumulate between the two, versus in sympatric speciation, genetic differences are going to be at the forefront, are going to happen first, okay? All right, so today you're going to have a few homework assignments, so check those out and let me know if you have questions. We'll have exam number 4, I guess it is, on Monday, same as what we've been doing. Let me know if you guys have any questions on any of that, and I will see you all next week. Bye.